Good morning. My name is Jim Pfaendtner, and I'm a professor of chemical engineering and the department chair, as well as a professor of chemistry at the University of Washington. And on behalf of James Ludwig, who's a PhD student in the Department of Chemistry, I'll be presenting today on new strategies for capture of uremic toxins from molecular simulations. Before I begin, I want to just briefly acknowledge my PhD student, Josh Smith. He graduated last summer, and he's now a data scientist at Just Biotherapeutics. The bulk of my presentation today will be slides from Josh's PhD defense, as well as research from this paper that's published here in case you're interested in looking at the reference. I'd also like to acknowledge financial support from the Center for Dialysis Innovation, funded by the Northwest Kidney Centers, as well as all the great scientific conversations and advice I received from Buddy and Jonathan, the directors, as well as Professors Bruce Hines and Daeong Gao. So many of you may know that kidney dialysis successfully treats only a portion of uremic toxins. All of you know that if left untreated, chronic kidney disease leads to the accumulation of life-threatening toxins in the body, as shown here by this cartoon on the left, with uremic toxins signified by these small little symbols, squares, and circles and triangles. So as kidney function decreases, eventually the level of toxins in the body exceeds some safe level, and people begin to experience significant health effects. Treatment options for CKD are rather limited: kidney transplantation and dialysis. Whereas kidney dialysis is hailed as a miracle lifesaver, and it truly is, many people are not aware that dialysis really only functions on removing urea and urea clearance from the body. Many other uremic toxins, such as the ones I'll be talking about today, are unable to be cleared with traditional dialysis methods. And so over time, chronic health effects do build up, and they do begin to cause more and more effects for patients. So the subject of my talk today are the so-called protein-bound uremic toxins, or PBUT, and they're shown here as this little cartoon of a protein signified by this small heart, and then a protein toxin complex with a small circle. So in a traditional dialyzer, these molecules are one large complex, and they wind up being far too large to pass through a membrane wall and leave in the dialysate, unlike urema, which exists as a small molecule and is able to be readily cleared through dialysis. So clearance of PBUTs remains a grand challenge in development of new dialysis technologies. Most PBUTs, to my knowledge, are travel through the body on universal transporter proteins. I've shown here in cartoon relief a protein called serum albumin. Serum albumin is a promiscuous transporter protein. It can bind many types of different small molecules. I'm showing here one uremic toxin, indoxyl sulfate. Indoxyl sulfate exists 85% or more bound to this protein in the body. And so even though it does cause health effects in its small molecule form, it moves around through the body when bound to this protein. When these proteins, which are in your blood, enter the kidney, there's mechanisms of release inside the kidney, which remove these toxins very quickly, freeing those molecules for clearance by the kidney, and importantly, freeing the transporter proteins for release for other tasks. So failure to remove these PBUTs from the transporter proteins actually causes a cascade of problems and chronic health effects. First of all, the level of uremic toxins continues to increase, but also an underappreciated health concern is that the transporter proteins themselves are unavailable for other important tasks in the body. We would like to develop new technologies to remove these transporter proteins, but fundamental knowledge about this release mechanism, precisely how the kidney is achieving really rapid release of these toxins, is lacking, preventing us from using a rational design approach for designing new treatment strategies. We're strong proponents in my group of mathematical modeling, and we think that we can build on some work that's already been done in the literature to help drive the field towards next generation dialysis for PBUT removal. One example of such modeling work is a paper here that I'm reporting from the group of Peter Kotenko. The cartoon diagram here is a so-called 3-compartment model for dialysis. And the authors of this paper presented a novel type of PBUT removal dialyzer. And by doing careful mathematical modeling of the actual dialyzer function of that device, they were able to show results such as that shown in the lower right-hand corner, that show that indoxyl sulfate concentrations in the body can decrease to safe levels over a reasonable timescale. Here you're seeing, for example, a 6-hour timescale of dialysis. So that's potentially very exciting, just showing that the basic mathematics of PBUT dialysis could be successful and that we could achieve PBUT removal with a dialyzer. But one thing that's really not addressed here is sort of this grand challenge of how to engineer the dialyzer. What goes inside that little blue box in that schematic? And really what we're trying to do here is beat nature. The serum albumin protein has evolved over almost millions of years in order to be incredibly efficient at binding small molecules and moving them around an organism. And what we're trying to do is figure out how we can beat that. We need to design materials that could actually capture PBUTs at a higher rate in many cases. So our approach is to develop a molecular or nanoscale understanding of serum albumin with uremic toxins or PBUT interactions, especially the release mechanisms. We believe that this will provide a wealth of new knowledge for helping material scientists and bioengineers to develop the next generation of dialyzer technology. So going down from the device scale, we need to move beyond cartoon level understanding. At the top of the slide here, I'm showing a classic, what I would call biochemical textbook view of a protein ligand binding problem, also a protein toxin binding problem. So we have a diagram here that looks like Pac-Man on the upper right, and we always see these simplified reactions in which a ligand or a toxin leaves the protein that it's bound to. These diagrams are simple and clear to understand. They're also amenable to sort of mathematical modeling approaches that don't involve the molecular scale. All of the interesting detail about the molecules and the nanoscale is encapsulated here in these coefficients. So the devil's really in the details. We have to move beyond this Pac-Man description down towards something that's at the nanoscale in order to really understand what's happening. So here what I've done is zoom way in on the serum albumin binding pocket, the so-called Sudlow binding site, where indoxyl sulfate, shown here in cyan and yellow, is really being held in place by the amino acid side chains that are colored blue and yellow. So those are parts of the protein that have strong chemical interactions with that toxin and lead to very strong binding. And just to give you an appreciation of the length scale that we're talking about here, the bar that I'm showing you is about 1 nanometer in length in terms of this atomic representation of the binding pocket. And for reference, a human hair is on the order of 10,000 nanometers in length. So we are truly talking about the size scale of molecules, and it's just important to remember, and I think it's always impressive to imagine at these very, very tiny length scales, the massive impact things can have in terms of healthy stasis of your body. This mechanism of ligand toxin release from a transporter protein is very challenging and incredibly costly to study with wet lab experiments. This could be years of work for a graduate student or postdoc. And so my group seeks to use potentially less accurate methods, but much more rapid methods for screening protein toxin interactions and for actually simulating them. So the method we use is called molecular dynamic simulations, and we are able to leverage supercomputers to do very large simulations of molecules, solvents like water, ions. And very briefly, the way this works is that we define a so-called energy function or force field that describes a molecule and its environment. And we simply then numerically evolve Newton's equations of motion, force equals mass times acceleration, on a supercomputer. So these simulations sometimes run for weeks using thousands of processors. And we create a large ensemble or a huge data set that then requires some detailed statistical analysis to determine things like structure, thermodynamics, kinetics, and ultimately the mechanism of unbinding. So just as an example, I'm showing every grad student's favorite molecule, caffeine. And on the right here is a small movie or cartoon of a molecular dynamic simulation. And the main thing I want to impress upon you, you can see the shaded line on the left kind of changing as a function of time. We're tracking how many water molecules are associating with caffeine, and it's rapidly changing over time. And so any instantaneous view of the molecular dynamic simulation doesn't represent the average behavior you would see in real life with a real microscope. But over time, we observe what's called this ensemble average, and we're able to calculate all sorts of interesting thermodynamic and kinetic quantities using those statistical averages. I also want to impress upon you that these simulations of biochemical processes can generate huge amounts of data. So the system has, hundreds of thousands of actual degrees of freedom or dimensions, and we're taking millions of observations over the course of a simulation. These simulations can take up terabytes of data, and so we also wind up doing a lot of really creative and interesting software engineering just to process all of this data. One of the first things we did was to begin to ask the question, well, what is known about protein-bound uremic toxins from actual wet lab experiments? And we found that there was only one measured toxin transporter protein binding site that was indoxyl sulfate. But there's many protein-bound uremic toxins that travel through the body in this binding site. P-cresyl sulfate, indole-3 acetic acid, and puric acid are examples. So we picked this panel of toxins to study for their chemical diversity, and also because they would represent a good opportunity to use our methods to predict the binding site of these toxins alongside indoxyl sulfate. Josh had this really creative idea to generate starting structures of these molecules by overlapping the new toxins with indoxyl sulfate, and then we launched large ensembles of molecular dynamic simulations and studied the stability of the protein and the stability of the toxins in the binding site over a long period of time. So one of the initial outcomes from this work that we're really excited about is now we have published structures, not of one, but of all 4 of these toxin protein binding sites that others could use for their own investigations and studies of the protein toxin interactions. Digging deeper though, we have the opportunity to really interrogate these systems and learn a lot about the key interactions, and again, all those details down at the nanoscale. And one of the really early observations that impressed us as we surveyed all of this data that we calculated from the supercomputer was the fact that, in contrast to the traditional dogmatic view of there being a single structure of a ligand or toxin in the protein binding site, that dogmatic view comes from the use of X-ray crystallography, which takes place at very low temperatures, near 0 Kelvin. In contrast to that dogmatic single site view, protein-bound uremic toxins are rather promiscuous. They tend to bounce around a lot inside the binding site, like I'm illustrating with this cartoon. So it's not as though Pac-Man's eating just a triangle and chomping down on it. There's lots of little nooks and crannies where that toxin might move around. So the analysis of all of these simulations and all of this data proves to be quite challenging. And we turned to machine learning as a way to take all of this data from the supercomputer and compress it down into representations like I'm showing here. This is a so-called Markov state model. It's a type of network that shows you where is the toxin within the binding site. And the size of the circle, for example, begins to tell us how likely is it that that toxin is going to be sitting, for example, here at site 5 versus this less likely position here at site 3 or site 4. Sorry about that. The next thing we do, and one of the other really important things we learn, is about the exact biochemical nature of the interactions with all these individual toxins with the local binding pocket environment. And we do this through an analysis called contact analysis. And the basic idea here is that we define the chemistry we're interested in. So I'm showing here again indoxyl sulfate. And the cyan and blue part represents that an indole ring that is rather hydrophobic in general. And then we have this anionic group right here, the sulfate group marked with yellow and red oxygen atoms. And we can then look at even at a length scale smaller than the whole toxin itself, we can look and see what type of side chains interact with this indole ring, what type of side chains interact with this anionic group, and then we go through and rank order them. And so I'm not gonna, of course, go through all the detail of these 4 or 8 box plots. There's one set for each of the toxins we looked at. And then there's a type of interactions that are more hydrophobic and type of interactions that are more anionic. So the cool thing that we can do is then begin to look across the series, this family of toxins that have diverse chemistry. We can ask the question, which of the interactions are prevalent across all of them, and begin to draw some more general conclusions. So when we do that, we can take all of these residues that are known to be near indoxyl sulfate in the experimental contact or experimental structure. We filter them according to these contacts that I just told you about. And what emerges that's really interesting is all of these chemical interactions with this whole panel of uremic toxins can be collapsed down onto 9 types of interactions. So there's different types of interactions that are listed here with the amino acid side chain and its residue number. There's polar ones shown in green. There's positively charged ones shown in blue, and then there's some apolar ones shown in gray. So this set of information is really useful, and it begins to tell us about the precise chemical nature of the average interactions or the structure across this diverse binding mechanism I told you about. Additionally, we can observe the mechanism. These are dynamic simulations where things are moving on the fly. So one piece of the mechanism that we found for indoxyl sulfate that was really interesting was this gatekeeper, where a residue arginine 410, which is positively charged, actually moves like a hinge up and down. And that movement allows indoxyl sulfate to have that promiscuous binding and travel around the binding pocket. So this plot here is quite interesting. It's time on the x-axis. This is the length scale of our molecular simulation in nanoseconds. And here's distance on the y-axis. And that's how far is the ligand from that arginine residue. And the coloration relates to energy. And so all you need to know, this is the energy of the interaction. Red is stronger and blue is less strong, or a weaker interaction. So what's really interesting is that we can track using the energy of the system when that side chain, the blue side chain, is close, it's lower in energy, and then as we move through, the side chain opens, that gate swings open, and then you can see it's very dynamic throughout the course of the simulation. On the time scale of 50 nanoseconds, that side chain is moving up and down, allowing the ligand to travel around the binding pocket. Similar results can be found for all 4 of the uremic toxins that we studied in this transporter protein, again indicating some sort of universality in the mechanism of capture of these uremic toxins within the indoxyl sulfate binding pocket. Those are just a few of the key results that we learned in this paper, and I again encourage you to find that paper by Josh Smith and me, Jim Pfaendtner, which was published this summer, if you want to know more. Some kind of high-level takeaways from my talk today are that with this grand challenge of developing new strategies for discovering dialyzer technologies for PBUT clearance, we see molecular modeling as a route to helping design those new materials, which are absolutely needed for the next generation devices that will go in those dialyzers for facilitating toxin release and capture of toxins. We seek to do this through accelerating the scientific pipeline with a technology called molecular dynamic simulations, a sort of computational microscope that allows us to learn fundamental mechanisms that have previously not been studied. And one of the really, I think, exciting conclusions that you can take away from my talk is that despite complexities in toxin binding, these PBUTs show common binding mechanisms across really diverse chemical spaces. So stay tuned for future research from our group and from the CDI. We're really excited to see how to harness this knowledge in the design of all sorts of new types of smart capture materials. Thank you so much for your attention, and for all of you researchers for your important work in advancing dialysis technology.
Thank you, Dr. Pfaendtner, for that stimulating talk. I'd now like to introduce Melissa West, who is the Acting Vice President for Research, Discovery, and Innovation at the ASN Alliance for Kidney Health, which includes KidneyX and the Kidney Health Initiative.